Good day. Ang pag-uusapan natin ngayon is all about microscope. Yung parts niya, function, at saka some history. Let's begin. Microscope comes from a Greek word micron, means small, and scopen means to look at. So, technically, pag sinabi natin microscope, ito ay isang device or instrument na ginagamit natin para mapalaki yung isang small object. So, para ma-magnify yung small object gamit yung mga different lenses. So, that's what microscope is. Bago tayo magtungo sa parts and function ng microscope, pag-usapan muna natin yung brief history nito. Yung timeline, and then ano ba yung compound light microscope, and then we'll go into the parts and function. So, ang timeline niya ay nagsimula noong 1665. So, ngayon ay 2021. Nagsimula siya noong 1665. So, roughly 400 years ago. Up to 1932, yan yung yan yung development ng microscope. Okay, yan yung mga taon na nag-involve sa development ng microscope. Although, syempre, ngayon, nag-evolve pa rin yung paggamit natin ng microscope. So, let's start with Robert Hooke. Sino ba si Robert Hooke? Siya yung nakadiscover ng existence ng cell. So, siya ang unang nakapagbigay ng word na cell. Okay? So, using a microscope, this is the first microscope na ginamit niya. Gumamit siya ng cork or yung takip ng wine. Kinuha niya yon and then tinignan niya under the microscope and napansin niya it is made up of little substance or little things. Ang tinawag niya yung cell. Next naman natin si Anton van Leeuwenhoek. Si Leeuwenhoek, in-improve niya naman yung ginawa ni Robert Hooke na microscope. Okay? So, siya naman ang nakadiscover ng cell from the bacteria. Or cell na gawa in a form of bacteria. And then, he discovered human cells, specifically sperm cell, and other animals as well. So, nakadiscover siya ng mga cell na ang cell pala or ang animals pala ay gawa din sa cells. Next, Robert Brown. He discovered naman yung sa loob ng cell, meron tayong tinatawag na nucleus. Specifically sa plant. Yun ang na-discover ni Robert Brown. Next, we have Matthias Jacob Sheldon. Siya naman nag-propose na lahat ng plants ay gawa sa cell. Meron tayong J.E. Purkinje and then Theodore Swan. Sino naman to mga to? Si J.E. Purkinje, siya naman nagbigay ng term na protoplasm. Ibig sabihin, ang protoplasm class, pag-aaral natin sa mga susunod na lesson, yan yung part na kung saan nakalutang yung mga yung nucleus at saka yung cytoplasm natin. Okay? Next, Theodore Swan. Siya naman na nagsabi na all plants and animals are consist of cells. So, hindi lang plants, kundi lahat, pati animals or living things for that matter is made up of cell. So, siya ang nag-establish ng cell theory. Next, we have Virchow. Siya naman nagpatuloy nung work ni Theodore na kung saan nirefine niya yung cell theory na kung saan binigyan niya pa ng much deeper meaning yung cell theory. Siya rin nagbigay ng idea na ang cell ay nanggaling sa iba pang cell. Next, we have Ernst Haeckel. Siya naman nakadiskubre ng hundred of species. Siya rin na nagbigay ng term na ecology. So, he popularized also the version of recapitulation theory during embryonic development of animals. Ibig sabihin, habang nade-develop yung animals from the embryo. Next, we have Max Knoll and Ernst Ruska. So, silang dalawa yung naging leader ng electron research group where they proposed the work of making an electron microscope, which is the most accurate microscope that we are using right now. Next, we are going to talk about the compound light microscope. Ano ba yung compound light microscope? This is a very basic type of microscope na kung saan gumagamit lang tayo ng simple lights and lenses to see an object, a small object to be magnified. So, ang compound microscope ay mayroong mga lenses, objective lenses. Typically, ang ginagawa nun ay sino-zoom in yung isang object 4 times, 10 times, 40 times, or 100 times. Okay? Meron din siyang part na eyepiece na kung saan imomultiply mo naman yun sa magnification niya. Merong magnification na 40, 100, 400, and 1,000 times. Okay? Then, the higher the magnification is achieved by using two lenses rather than a single magnifying lens. So, dalawa po yung lens na ginagamit. Yung eyepiece, which is nasa taas, and then yung nasa baba, which is the objective. To reach maximum capacity or capability na makakita tayo ng small object. Okay? While the eyepiece and the objective lens create high magnification, the condenser beneath the stage focuses the light directly to the sample. So, hindi natin basta-basta makikita yung specimen not unless meron tayong light. So, meron ang nagko-control ng light sa ilalim ay yung condenser or also known as diaphragm. Okay? And then the light source is the mirror. So, binabounce lang ng mirror yung light papunta dun sa ating specimen. So here are the basic parts of the microscope. So meron tayong eyepiece, body tube, coarse adjustment knob, fine adjustment arm, inclination joint, condenser adjustment, mirror, condenser or diaphragm, stage objective, and foot or base. Okay. So, here are the parts of the magnifying parts. So, meron tayong eyepiece, kaya nasabi ko kanina, and then objective. Okay? Meron tayong 4 kinds of objectives. We have the scanner, low power, high power, and oil immersion. Usually, ang scanner, 4 times magnification lang. Low power, 10 times magnification. High power, 100 times magnification. Oil immersion naman, umaabot ng 100 times of magnification. But kailangan natin gumamit ng oil para mas lalong maging clear yung specimen natin. Next, ito naman yung illuminating part. Ano naman yung illuminating part? Sila yung nagpapailaw ng ating specimen. So, meron tayong diaphragm, controls the amount of light reaching the specimen. Condenser focuses light on the object. Then, mirror. It reflects the light passed through the hole in the stage. So, siya yung nagiging source ng light natin ng microscope. Next, we have the mechanical parts. Ito naman yung mga ginagalaw na parts ng microscope. We have the arm. So, arm para mahawakan ng maigi yung microscope. Base, siya yung pinaka-base ng microscope para makatayo. The body tube, siya naman yung nagko-connect ng eyepiece sa objective natin. Next, we have the coarse adjustment knob, fine adjustment knob, at inclination joint. Ito yung part pa rin ng mechanical part natin. So, coarse adjustment knob, ito po yung knob na pinakamalaki sa microscope na kung saan pinipihit siya para magalaw yung ating body tube at makita yung, mapokus natin yung specimen. Now, we have the fine adjustment tab naman. Ang fine adjustment tab ay ginagamit kapag na-focus mo na yung specimen pero medyo malabo pa, kailangan mong gamitin yung fine adjustment para mas lalo siyang mapalinaw. Next, we have the inclination joint. Inclination joint ay nakakonect sa base and the arm, in which you can tilt it or pwede mo siyang galawin para maging komportable ka sa pagsilip ng microscope. Next, we have the nose piece, stage, and stage clip. Again, this is this is still part of the mechanical parts of the microscope or the moving parts. We have the nose piece. The nose piece, siya po yung nag-hold ng ating objectives na kung saan niro-rotate mo siya kung anong gusto mong gamiting objectives. Gusto mo ba yung 4 times, 40 times, 100 times, or 10 times? Yan po ang rotate no ating objective. Then we have the stage. Ito yung flat surface na kung saan doon mo nilalagay yung ating specimen. Okay, yung slide. Then stage clip, meron pong clip yung ating stage na kung saan ang silbi nun ay para hindi gumalaw-galaw yung ating specimen or yung ating slide. So that's all for today, class. We will going to discuss further about this by next meeting. See you again next meeting. Goodbye.